Hey, hello, welcome everyone. I'm Trent Blizzard with NAMA, and I am here with Harte Singer. Say hi, Hart.
How's it going?
All right, Hart's going to be our webinar presenter tonight on DNA to Z, Adventures from the Front Lines of Biodiversity Discovery. So we're pretty excited about that Hart, and I want to welcome you. And I want to do a couple of quick introductions and talk a little bit about you, and then I'll turn it over. How's that sound?
Sounds great.
All right. Well, we've got a couple of big forays coming up. You'll want to check out the Similkameen Valley Foray and Feast in BC in early June. Just opened registration. Our big annual foray is in Marquette, Michigan in the end of August. That's the NAMA the 65th annual foray. And we have our foray going to Oaxaca City, NAMA MX 26, and that's going to happen September 6th through the 13th. So check out our forays. Hope to see you on one of them. We've got some webinars coming up. I see here we have Graham Steinreich presenting, I guess that's this Thursday, on community science and mycology, documenting fungal diversity through collaboration. That'll be a pretty good one. Are you gonna listen in on that one, Art?
Yeah, actually, Graham was just staying at my house a week ago, so this will be a good lead up to that. I touch on community science just a wee bit, but I'll leave the rest of it to him.
Excellent. Dr. Amy Honan, Wings and Waste, How Fungi Get Around is coming up February 23rd. Zachary Mazzi, Areri Monteiro, Kim Hunter, and Dave Layton are presenting on Shared Purpose on Sacred Land, the Mushroom People of the Oaxacan Mountains, and that is February 25th. So that's our February schedule. We'll I think move on from there. And remind everyone we do have a logo contest going on right now for our annual foray if anybody is interested in competing in a logo contest. So here we are with Hart, and I've known you for a few years now. Now, Hart, I think I met you at Soma a few years back, and then more and more we see each other at NAMA forays because you're so involved with the DNA side of NAMA. You're one of the founding members of the DNA committee, and you're helping a lot through the volunteer sequencing efforts. So I want to thank you for all that you do with NAMA and all the people you help in the organization and kind of what you represent. You do so much for the organization. Thank you.
So thank you so much. Yeah, the pleasure's all mine, and the opportunity wouldn't be there without you guys. So I appreciate it.
Okay, well, I'm gonna say, why don't you start up your screen here, and I'm gonna step out, and I'm gonna sit back and watch, and I may pop in and ask a question or two along the way.
Yep. So while we get started, this is a QR code here. If you want, you can access the slides that I'm using. It's just a link tree QR code. So there's past slideshows and links to all my protocols and other things. So feel free to do that. If you have questions and they're burning questions, that can be answered fairly quickly. Go ahead and ask them. And Trent has a secret way of letting me know that you have a question, and I'll answer it. If you've got a little bit more long format questions, then it would be great to save them to the end. I'll try to make sure there's plenty of time to go over them. All right. So let me get started. And before I tell anybody about myself, I wouldn't be here without some pretty incredible people that were patient, mentored me, and were generous with their time. So the first gentleman is Damon Ty. I'm sure many of you know this guy. He's sort of a cornerstone of the community, not just in mycology, but in community science in general. And would not be here without Damon. Wouldn't have met all the next two people in line without Damon. So this gentleman here, Alan Rockefeller, also pretty well-known face in the community. Here's him at Soma Camp in 2020 with a very apt shirt. And Alan also took a lot of time to show me how to do lab work, PCR, DNA extraction, microscopy, a lot of these things that at the time I wasn't a student or an academic at all. So big thanks. And then finally, when I did breach through the threshold into academia, this gentleman here, Brian Perry, who's my thesis advisor, has been instrumental in supporting me and supporting the Fundus project. We wouldn't be able to do what we do without him. So, got to get that out of the way. Big thanks to these folks and many others who I couldn't fit on one slide. I could probably spend an entire presentation talking about all of the people that hold me up in this world. So let's talk a little bit about me. I got into mushrooms because I had a cooking career. So that picture of me there is 20 years ago when I was just learning to enjoy mushrooms. I grew up hating them. I thought their texture was horrible. It turns out if you learn how to cook mushrooms, they can turn out pretty good. And so I really loved cooking mushrooms. And then a little bit later on, I quit my cooking career. I wanted to move on in my life. I actually had time to forage for mushrooms. So this is my first time finding mountain porcini in California in 2018. They're kind of a hard thing to come by. They occur after sporadic summer thunderstorms. And so I didn't even know these were going to be up there. It was one of the best days of my life. And nowadays I do DNA sequencing. And so we'll find out a little bit about what that is as I go through the slides. I am a board member of Bay Area Applied Mycology, the Entheum Foundation, and Sonoma County Mycological Association. I currently attend Cal State East Bay, although barely. I'm just finishing my thesis work to graduate with a master's in biology. I'm one of the founding members of the DNAMA DNA Sequencing Committee. I previously had a paying job doing what I do for the Fungal Diversity Survey, which was an incredible story. I wish I could had time to tell you all about it. But and then finally, I also run a very, very small business operation out of my garage doing DNA barcoding work. So this is not a sales pitch, but if you're ever interested, there's someone who does it. Okay, I want to talk about Fundus briefly. Because this is kind of how I got my footing in the world of doing DNA barcoding. So Fundus, short for Fungal Diversity Survey, and we have a vision in which a world in which the fungal kingdom is fully documented, protected, understood, and appreciated. Who is Fundus? A group of community scientists who share that vision. So just a few examples of some exceptional people that work for Fundus. Here's Rudy Diaz out of Los Angeles Mycological Society. That's Tay Bright. She's a co-board member with me at SOMA and also collected for Fundus. Stu Pickle, another exceptional dude in Los Angeles. And this other guy over there. One thing we have in common is we really like to get all up and close and personal with the mushrooms and give them a good sniff. Okay, I use the term community science. I'm going to define it briefly. I'm going to leave the bulk of the discussion of that up to Graham Steinruck, who you'll see in a week or in a few days. Okay, this is sort of how I like to describe community science as the intersection between amateur and academic communities. And this picture really describes that scene. So Josh Berkeback over there, he's the chair of our DNAMA committee. Kyle Kanan, who runs the Ohio Mushroom DNA Lab, and Scott Ostuni, the first sequence validator ever hired in history, who now is busy describing new species left and right. Here we are in an Airbnb in Alabama in 2023, I believe, yep, for the Alabama Mushroom Fair. And we are doing nanopore sequencing right here in the Airbnb and having a good time doing it. So community science is science that transcends the boundaries of institutions, and it's often conducted by amateurs, not always. An amateur is from the Latin word for amator, one who engages in a pursuit, studies, science, or sport as a pastime rather than a profession. Also, one lacking in experience and competence in an art or science. let's abandon that definition. We don't like that. Let's go back to the root of this as an amateur being a lover, someone who is doing something purely out of love. The community science glue that sticks a lot of us together is the DNAMA Sequencing Committee. As I mentioned, the chair is Dr. Joshua Birkeback, a clavariologist extraordinaire and a righteous leader. In our committee, we have some notable members. Stephen Russell, a NAMA OG, founding member of Fundus, director of Mycota Lab, and Mycomap Network. Jessica Williams, Fundus sequence validator, the second one that I hired. Also validated sequences for OMDL, fungarium technician, Dr. Andrew Wilson, who's been a longtime NAMA contributor and takes care of the collections at the Denver Botanic Gardens, but is also intimately involved in our specimen to fungarium pipeline with the Field Museum. Kyle Kanan, director of OMDL, and Scott Ostuni, I spoke about him a second ago. So what does the DNAMA committee do? We're like the central point where all the separate labs convene and come up with really cool ideas and execute them. So here's one, I can't remember who came up with this, but this is the commercial mycoblitz. This is one of the DNAMA projects that was spearheaded by the Ohio Mushroom DNA Lab, and it was a really cool idea. So wild mushrooms and mushroom products are a $45 billion annual global market. In the United States, there are very vague labeling requirements, and it's difficult to put labels on these because often common names are obscure and don't translate well between cultures. And then scientific names change all the time. I'm sure everybody is a huge fan of that. So we thought, well, why don't we try to see in all of these commercial products if the names they're applying to them actually match the material inside of the containers. And in some cases, that was a little bit harder than expected. So the volunteers submitted 235 mushroom products to Ohio Mushroom DNA Lab for barcoding. The majority were fresh or dried, but some were also pickled and canned, and others were medicinal products like coffees and powders. Many of them had no country of origin. So, they were just sort of random mushrooms in a bag from who knows where. Some of them had no label other than fungus or mushroom. So especially in these times when we have a quote unquote epidemic of mushroom poisoning in California, this seems totally insane that you can just go to the store and buy a bunch of like random unlabeled mushrooms from an unknown location. And you're supposed to trust that they are good to eat. We don't see a lot of evidence of mushroom poisoning coming from these commercially sold mushrooms. So I don't think it's as big of a problem as it appears to be, but it'd be really nice to know what species you're eating when you're buying a bag of mushrooms. For example, most of the samples that were labeled porcini were actually just Swillus luteus, which is a perfectly good edible mushroom, but not a porcini. So once we did the DNA barcoding, or rather that was Kyle, there was the analysis process. So that was turned over to a sort of subcommittee of the DNAMA sequencing committee, which is called the Volunteer Sequence Validation Corps. So kind of like the Peace Corps, not like the Marine Corps. So this evolved from my first volunteer position with Fundus, which I was graciously handed a bucket of sequences by Dr. Biddy Roy and Dr. Jean Lodge, and trusted with the task of identifying them and uploading them to NCBI GenBank. I was quite honored that was handed to me, since I really personally didn't think I knew what I was doing, but I had proven myself in some way or form to the mushroom gods. So I had a group of people look through those sequences. We sort of established some protocols, and then just at the tail end of that, I got extremely lucky, and Fundus got a large grant from the state of California to survey mushrooms. So we did that. And then after the funding dried up for that, it became a hobby again. And I reignited this volunteer sequence validation team. So it's just any group of volunteers that want to join. In the slides, there's some links here, so you can click that, and that would bring you to this form where you can decide you want to join. And then there's a link here, which brings you to the guidebook. This is from July. It's quite out of date. The development of software tools and protocols is running so fast that my typing fingers can't keep up with it. But this will be a great starting point for you to learn how to look at DNA sequence data. And then if you decide you want to do this, I also have a public meeting calendar. So about 6 times a month, I hold meetings where folks that are interested in learning how to do DNA sequence validation can join and learn. And so it was a group of these folks. You can see them listed here. We got, in this column, Kevin, Kurt, John Plischke, some pretty heavy hitting names here. These folks helped look through all the sequence data, and we, for example, would look at these Morchella and identify them. This Flammulina came out as a hypsizygous. So we were able to sort of get more realistic names applied to these sort of random bags of mushrooms. There is a draft publication for this, which is being submitted to a very new journal called Marilia. And so look forward to seeing this published. Really, I didn't do much work on this at all, other than sort of put my team of volunteers on the sequence analysis part. You can really thank Kyle Kanan for doing most of the work and the volunteers who collected the samples. All right. So I've thrown this term around: DNA barcoding, DNA sequence validation. Let's get to the bottom of this, and I'm going to do this quickly. I could give an hour and a half presentation just on this alone because there's a lot to it, but I'm gonna keep it short for you guys. If you have questions about this, save those for the end because I'll probably answer in detail. Okay, let's get to some basics here. DNA is a versatile molecule that makes up the instructions for life. So every protein in your body, that starts out as a DNA sequence. A DNA sequence is basically a combination of 4 letters. And here's the 4 letters in fancy chemical format: adenine, thymine, guanine, and cytosine. And here's another graphic showing the DNA double helix and then what's going on the molecular level in there. So there's a thing called complementary base pairing, where thymine always pairs with adenine and guanine always with cytosine. The long form name of DNA is deoxyribonucleic acid. So all of the DNA inside of an organism is what we call the genome. So you've probably heard this term genome or genomics, whole genome sequencing. This is where you sequence every single DNA letter in the entire genome. If you're a human being, which most of us are, you inherit 3 billion DNA letters from each parent. So that's a lot of DNA to look at. If you're a mushroom, and some of you might be, you probably have about 50 million DNA letters. So orders of magnitude less DNA, as mushrooms are a little bit less complicated than people. None of this is going to be helpful for DNA barcoding. It's going to take a long time to analyze giant data sets of millions and millions of DNA letters. So what we do when we want to identify what mushroom is in a random package from a supermarket, we go for what's called a DNA barcode. It's a tiny portion of the genome that's used like a barcode, like you would scan at checkout, at the self-checkout, to identify a mushroom. Now, I should caveat this with the idea that whatever entity created the universe didn't, for our convenience, place barcodes inside the genome. It's what we would call a heuristic, or a sort of little shortcut that isn't perfect. Nothing in nature is. So you want to take this idea of a barcode with a grain of salt. You can't just always get a barcode and have a clean and simple identification. We'll get into more of that in a little bit. What a barcode sequence would look like is this right here is the fungal ITS barcode. I don't know what specimen it came from, but that's about how big it is. I would say on average 600 to 700 DNA letters, or we like to call them base pairs long. How we get the DNA barcode is a process that you might be familiar with called PCR. But before we do the PCR, we start by extracting DNA. And we have gotten our DNA extraction technique down to a science, literally. So what I do is I fill up a bunch of tiny little tubes like this one over here with a buffer. And a buffer is just a solution that maintains a constant pH. And inside that buffer, there's a couple of chemicals that help break down the components of cells, like cell walls, cell membranes, and some ions that help the DNA escape from those cells. So what we do is we take a standard issue toothpick, unflavored ideally, individually wrapped is even better so you don't cross-contaminate, and we take that toothpick and rub it on the hymenium, or the fertile surface of a mushroom. And that gets a lot of these generative cells. So in a basidiomycete like this on the gills, you've got cells called basidia. And there's so many of them, and there's a lot of nuclei in those cells that are dividing and undergoing meiosis to form spores. So you have a whole ton of DNA going on the surface of the gills, or if it's a cup fungus on the surface of the epithecium, etc. Wherever there's a generative material, you're going to have a lot of DNA. So just scratching that with a toothpick, even to the point where you can't see any material attached to the toothpick, is enough to capture enough DNA for us to get a DNA barcode. So the next step is PCR. And since we've been through COVID times, everyone's heard the term PCR. But what it means is polymerase chain reaction. Polymerase is an enzyme that copies DNA, and it's a chain reaction because it happens in an exponential manner. So you start out with one double-stranded copy of DNA. You melt it by using heat, and that separates it into two strands. Then you add these things called primers, and the primers basically define that region that you want to amplify, which is the barcode. And then you copy it with an enzyme. And this all happens just by changing the temperature a few times. Of course, we add the ingredients, the polymerase, primers, and the individual DNA letters, and those are the things that actually do the magic. But the act of doing PCR is just putting it in a machine that changes temperature over and over again. It's exponential, so after I usually do about 32 cycles, you have 2 to the 32nd copies. I don't have a calculator handy, but that's it's like billions. So what we've done with PCR is we've basically identified a region that we want to amplify, and we amplify it to the point where so much of the DNA is just that region that the other DNA doesn't really matter. So the other 50 million DNA letters in the fungal genome are drowned out by this copies of this barcode. Then what we do is we sequence. And so the sequencing takes place with this really crazy technology that I'm not going to try to explain to you because I found a really cool video on the website that shows it. And that'll be the next slide. And it's got crazy audio, so enjoy. But we'll take that DNA and we will use some crazy bacterial proteins embedded in a electronic device to sequence it. So without further ado, I will present the nanopore video of how the sequencing technology works. Get ready to party. If you can't hear it, let me. That's the DNA. Cool. So, if you have questions about, go ahead.
Hi, Hart. We got a couple of questions here I wanted to ask you.
Yeah,
Um,
go for it.
Barati and Rose are asking about the DNA from the wood on the toothpicks. Is there an easy way to weed that kind of thing out? Or other contaminants where you scratch the toothpick, i.e., herbarium mold, etc. So toothpicks and contaminants. Can you talk about that?
Yeah, absolutely. So, the nice thing about doing PCR to amplify the DNA that we want is we can use the primers that are specific to fungi. So they won't amplify the DNA of the wood, so you won't get the tree sequence. And I've never had a situation where we amplified a contaminant from a toothpick. I think they don't use moldy wood to make toothpicks, hopefully, ideally. And the way they manufacture them must be in such a situation that we don't end up with contaminants. And later on, I'll maybe show a slide of a situation where I've done PCR in a very dirty environment and not had any weird contamination. So it actually works really well. And I think I'm not the one who have come up with this. I think it was the Matheny lab that developed this technique.
Another question is how from Charlene is how do we access the slides that have clickable links for the guidebooks?
Oh, so when you go to that QR code, does it? Yeah, that's right. Because you probably have to. Let me get back to you on that one. Let me make sure that it works. I'm not going to do that right now, but after this, I'll make sure that there's a way for you to click on the links.
Excellent, thank you. Rose asks, are there different primers for different kinds of fungi?
Yes, although for what we do, we use what's called a universal fungal primer, and that's misleading because it only amplifies 90 to 95% of fungi. You won't really get some lower fungi like chytrid fungi won't amplify with it. But for different groups of fungi, you can get very specific primers. You can get ones that only work in basidiomycetes. So there's a lot of customization.
Are there any species that are especially hard to sequence?
Yes.
Barcode.
With our standard primers, we can't get chanterelles. I wouldn't say we can't get them. It's that we don't generate sequences very often using these standard primers. And there's so little reference data of this region in chanterelles that typically one would amplify an entirely different region. Um, there's certainly examples of things that don't work well. So I wouldn't say this is a magic tool.
Thanks. I'd say, Scott asks, how do you use a toothpick on a dried specimen?
So that's a great question. I haven't attempted that much. We did it inadvertently at NAMA in Vermont because a lot of the specimens were just naturally dried. It was quite a dry year. I'll get into that in a little bit. But I think you can just do the same technique where you scratch the gills or the fertile surface. Stephen Russell has done some tests with that, so he'd be a great person to ask.
Okay, I'd say thanks and continue on. I'll come back later with more questions for you.
Awesome. Thank you so much. Um, so we discussed the mechanism by which we generate a DNA barcode. All of that is so interesting, and I'm happy to chat more about that after this. But why do we generate a barcode? I mean, it's not just to identify mushrooms that you find at the supermarket. There's got to be a better reason for it. And the main reason is that mushrooms are hard to identify. These two mushrooms that you've been looking at for the past few minutes, if I was in the field and I went and saw some pink mushrooms fruiting out of the snow in the Pacific Northwest in the Cascade Mountains region, I would consider them Hygrophorus getzii. It's one of the fundus rare challenge mushrooms. And in fact, these two collections were made not far apart from each other near Crater Lake, growing with similar host trees, both growing from snowmelt. So they were both identified as Hygrophorus getzii, and they were sent to me to do sequencing. However, as you can already tell, there's a bit of a difference between these. They're not the same. So this mushroom here, we were able to identify as Hygrophorus vernalis PNW14. And that name may sound a little bit strange. It's what we assume to be an undescribed species. It does not match our reference sequences for Hygrophorus vernalis, but it's close. It's a little bit more than 1% away, but it's more than 5% away from Hygrophorus getzii. These are subtle things that you wouldn't be able to tell by looking at it in the field, but generally, Hygrophorus vernalis PNW14 has a more brown tones to it. And this one has pinker tones. And, maybe I can see that, but it's really hard. These names we generate ad hoc to sort of be able to collect these potentially undescribed species into the same bucket for analysis. So every time we sequence something that matches this, we can give it this, what we call temporary code name or cryptonomen temporarium, as Rod Toulos designated it. Other folks might use the term provisional name. However, provisional usually implies that it's the name that will be used for a description. And while some people have certainly used temporary code names in their description, like the recently described Gliophorus calunis, which was Gliophorus species California 01. Other folks might want to actually come up with different names. So we wouldn't describe this as Hygrophorus vernalis PNW14. So that's one reason we want to do barcoding. Mushrooms are hard to identify. A lot of historical collections were described many times, one species. I think Alexander Smith described one species of Hebeloma like 25 or 35 times. Don't quote me on that. But a large number of times because there were subtle differences in it and it seemed like it was new. And maybe the micro characters were slightly different from one group to the other. But with DNA barcoding, we can show that they are, in fact, the same. To get a little bit more philosophical, mushrooms are the ephemeral components of the fungal life cycle. They only exist temporarily for reproductive purposes. The mycelium that exists underground or in the wood or whatever substrate the mushroom or the fungus lives in encounters vastly different evolutionary pressures. So, for example, a mushroom that's in two different types of soil with two different soil communities, different plants around them, different bacteria, may have vastly different evolutionary pressure on the mycelium to produce different compounds to help defend against bacteria or other fungi, or to exist and digest components of the soil in one location versus the other. Whereas the fruit body, its job is to spread spores. It doesn't care what the soil conditions are like for the most part. It's not concerned with that. Its job is to pop out and disperse spores. So it has a different set of evolutionary pressure acting on it. So the mycelium may diverge into different species faster than the fruit bodies may change in appearance. DNA barcoding provides an additional piece of evidence. Do not use it as a silver bullet. The universe did not include a barcode in the genome for our convenience. As I mentioned before, it's a heuristic, and in many cases it does fail, but in most cases it works. So it works best when we pair it with good photos, geolocation, and habitat data. And so being able to do DNA barcoding at an event where we have the physical mushrooms undried and collected nearby gives us a lot of those characters to pair with that DNA barcode. So I'm going to shift to now talking about the barcoding work that we've done at NAMA Forays. And so a little bit of history. The first efforts were by Stephen Russell when he was part of the AIM lab to sequence NAMA collections that were at the Field Museum. And this happened in 2014. The first NAMA foray that had plant sequencing, where Stephen and et al. Actually collected specimens at the foray for sequencing. That was in Cable, Wisconsin in 2017. And the first on-site sequencing happened in Randall, Washington in 2024. So that was where not only were the mushrooms sampled, PCR'd, all these things happened, but the sequencing also happened at least partially on-site. During that event, we were able to generate 800 DNA barcodes on site, and then Stephen Russell and Mycota lab sequenced everything that we missed in our first pass. So we ended up with more than 1,200 DNA barcode sequences from that event. In total, between this and all the previous events and all the other NAMA-sponsored events, we've generated 6,394 DNA barcodes that have Mushroom Observer or iNaturalist records. So that's a pretty substantial number. Let's talk a little bit about NAMA 24. This was my first experience driving my lab gear out somewhere and setting it up and trying to create a lab in situ. It's not my first time creating a lab in situ. I actually did that in Hawaii the year prior with Graham Steinruck and the Biodiversity Collective. But this is the first time driving it to an event and really setting up for large scale, high throughput barcoding. So this is what the lab table looked like. A couple of PCR machines, our spinny thing, whatever you call that, computer, and other equipment, necessary beverages. And over here we have two assistants that were there. This is Ryan Peace. Ryan works with Mycota Lab, and actually is, if you've done sequencing with Mycota in the past couple of years, this gentleman has been the one to do the sequencing of your specimens. This is Mandy Cork, a strong advocate for community science, and also I think currently in Ecuador with Alan. This was a really big space we had at NAMA 2024. So mushrooms are all laid out here. I'll get into the sort of nitty-gritty of the process in the next set of slides. But I was able to do all the PCR on site, and then I loaded the sequencer on the last day and packed it in my car and carefully sort of, sandwiched it in there and drove back to California from Washington with the sequencer running. So it's kind of cool. At Randall, Washington, we published the results on bioRxiv, so it's not published in a journal, but nonetheless, you can access them via a DOI. So if you click that, it'll bring you here. And Joshua helped greatly by putting this together with his expert PhD touch. And so I encourage you all to look at this. But here's a summary of the results. We did 801 successful sequences with a success rate of 82.7%. From that, we had 9 new records for the state of Washington. And even more exciting, 18 records that had never been sequenced. So I want to be careful with my use of language here. I'm not saying there's 18 new species that we discovered. Although I strongly suspect most of those are new species, we can't really say that they're all new species because we don't have a reference library of every mushroom that's ever been described. If one of these matched to a holotype collection of another mushroom, we could say, oh, okay, this is that species. But most holotypes have not been sequenced. A lot of holotypes are buried in old museums, and they were collected, over 100 years ago, many of them in Europe. And in some cases, they don't even have any physical material left. A lot of holotypes from really old school days are just drawings. And in other cases, the actual mushroom material has broken down or been consumed by insects, or otherwise the DNA is so degraded that it would cost quite a lot of money to try and get a barcode sequence from the holotype. So I use this term never before sequence taxa, or new sequences for short. So when I say new sequences, I'm saying hint, hint, possibly, probably new species, but with a caveat. The new sequence discovery rate for this event was 2%. So that doesn't sound like a pretty big number, but to think that just from one weekend, we were able to find what might be 18 new species, that's pretty exciting. You don't get that in other realms of biology. It's not like you can go on a camping trip with 300 people and find 18 new species of animals. Maybe if you were going to the tropics and you had a bunch of insect nets, maybe you'd find 18 new species of insects. But with fungi, we have similar levels of discovery to be done. Cool. So this is a little clip from the publication. It shows some of the new taxa that we found, or potentially new taxa, new sequences. What you'll notice about these is that they're all pretty small, not this type of stuff that sort of the average person would find. It was raining really hard all weekend at NAMA 2024. And so the average person probably wasn't going to be crawling on their hands and knees, digging through the dirt to find obscure tiny mushrooms. So the majority of these were found by sort of professional mycologists. I think Connor Dooley found at least 3 of them. Looks like we might have a question.
Hey, we got a few questions coming in here, so I figured I should
go for
send a few your way. There's Sarah once asked for you to explain what holotypes are, and Eliza asks, could you tell us more about holotypes and how specific ones are chosen?
Oh, that's a great question. If I use a word that people don't recognize, feel free to ask, because I have so many words that I can't remember which ones are common and which ones are not commonly known. So holotype refers to the original collection of something that's used in the description. So, for example, if I wanted to describe this, the name is escaping me, not Phaeomerasmius, Flammulaster. We have this collection from NAMA, and perhaps I would designate this as the holotype collection. So I would do microscopy on this collection. I would take all my notes from this collection. I would measure it, photograph it with a color scale, do sequencing of a couple of different genes, so not just the standard barcoding gene. And so this would end up being a physical manifestation of the thing that I describe as a species. So that in the future, if someone wants to analyze the work that I've done or wants to revisit the specimen that the description came from, they can find the original material used. So it's like a hard copy of the species description.
All right, thank you. Heather Erickson asks, why is barcoding limited to spore surfaces? Wouldn't cap or mycelial tissue also lend themselves to DNA barcoding?
Great question. Yes, so you're totally right. You can use tissue from any part of the mushroom to barcode for the most part. It's just that the hyphae that make up the context of the cap or the stipe are generally these large, inflated or long, thin cells, and the nuclei aren't very concentrated in them. So in a long hypha, it's you're going to have two little nuclei in this really big long cell, or in the context of the cap, you might have this big inflated cell with a tiny little nuclei there. At the fertile surface, you have a bunch of cells that are rapidly dividing, and so there's a bunch of nuclei available and a bunch of DNA. But you can absolutely, if you don't have gill tissue or hymeneal tissue, you can sample from any part of a mushroom. And with the barcoding locus that we use, ITS, it generally works really well. So great question.
Okay. Krista says, when you were describing the nanopore process, did you say that there was a bacterial aspect to recording the sequence, or did I misunderstand? So within the nanopore cartridge, it's both a mechanical and biological process to get the sequence, question mark.
Yes, it's like a miniature RoboCop, except the biological component isn't a human. It's bacterial protein. Or at least a protein that originated from bacteria. It's a protein called alpha hemolysin, or at least originally the first paper about nanopore sequencing used that. And it's basically like a donut-shaped protein that embeds itself in a membrane, and you can squeeze a DNA strand through it. So it is this weird mixture of biological and non-biological in one device.
Okay. Well, this is the last question. It's a pair of questions relating to the supermarket study. I'll read them and then I'll sign off and you can continue. Mark asked, did you publish it? And Rick Vanderpool asks, what is the best estimate about accuracy of received specimens with IDs versus what the barcodes reveal? So thanks.
Great. We have the grocery store or the commercial microblitz paper put together. It has not been published yet. We've submitted it, and correct me if I'm wrong, if Joshua was on the call or not. I believe it's been submitted to a newer journal called Morelia in Florida, which has been used to publish new species descriptions. Publishing in the science world is often a challenging thing. Publishers are kind of a racket. They charge a lot of money. They want you to publish very specific things in very specific formats. And if you want to just get your information out there and you don't want to pay a whole bunch of money to the publisher, there aren't a ton of options. I know NAMA used to have a journal called Mycology, which may still exist in some form or another. I know Joshua has published in there before. But yeah, so it's going to be published. And the other question was, so correct me if I'm wrong, but how confident can you be that a barcode coming from something actually matches the material that you sent in? And that's what we have a team of sequence validators for. So you can't always trust the data that you get from a sequencing run because mistakes happen, cross-contamination happens. Maybe in your tackle box you had an ascomycete next to a basidiomycete, or maybe you had two species of Cortinarius right next to each other. So there is currently a pretty important human component into actually looking at the data, looking at a photograph of the specimen, looking at other context, and trying to really truth that out. When it comes to the grocery store stuff, we didn't really, a lot of these were just labeled fungus or had the wrong name on it. So, there is an error bar in the interpretation of those results that we can't resolve. So that's a great question. All right. So I'm going to move along. And just what I was talking about here at NAMA at Randall, I see, there's a hand raised for a question. I didn't know I could see that. Cool. I can't hear you ask it, but you'll probably have to ask it in the chat. Maybe let me get through this slide. We'll take another question break in a couple of minutes. So one of the things at NAMA 2024, it was raining, and we had a lot of folks that aren't experienced mycologists. So you're walking through the woods, it's raining, you see a big mushroom, that's going to be the thing that you grab and bring back to the ID table. The vast majority of the new species or new sequences were collected by experienced mycologists who were sort of digging through the soil. The one reason why we don't see a lot of records that were new to the state of Washington is because Washington is the fourth most sampled state for DNA barcoding. Puget Sound Mycological Society and Danny Miller and Yimin Wang and a couple of other folks, South Sound Mycology Club, all these folks do a lot of DNA barcoding in the state of Washington. So they've really saturated the state. We see similar stuff in California where we've sort of bent the accumulation curve of finding new things. And so what it really takes, the point of all this is that if you want to find the interesting new stuff, you got to get down on your hands and knees and crawl around. We'll get into that in a minute. We're going to transition over to this past year's NAMA 2025 in Marlborough, Vermont. This was the beautiful campus that we get to stay at. So what I'll do is talk a little bit about the process. So let me unveil what actually happened on a day-to-day basis, how we were able to execute this. So here you can see the main gymnasium room or auditorium rather, where we had everything set up. Up here on the top was the VCP, the voucher collection project. And so that was the folks that are collecting for the field museum. And then all the other specimens were sort of laid out on these tables. What you can't see is outside there were intake tables, so people would come back from forays and drop off specimens. So after they arrived from the forays, there would be local experts, mostly Noah Siegel, identifying the mushrooms. Once they were identified, then people from the VCP who were also experts, would look for specimens that were really great examples of a particular species or were collections of stuff that nobody recognized, which could be used potentially as holotypes in the future. And so we attempted to intercept mushrooms at various stages of the pipeline. The most important thing for us was to barcode everything that was going to end up as a specimen at the field museum. Because we know those are going to be preserved in their entirety, and they're sort of archival collections. They were carefully photographed and entered into databases. So those are the things that we really tried to go after. But then we also tried to get as many specimens as possible. It was kind of a dry year, so at first we were just going for everything. We were getting turkey tails and every polypore that came in. We soon realized that even in a dry year, you can find some interesting stuff. So we quickly shifted our approach to sort of ignoring some of the more common things and trying to seek out more interesting specimens. There were a lot of collections that came in. There were sort of two components. There was the sampling folks and then the photography folks. I'll get into that in a second. But with our sort of chaotic and planned on-the-fly approach, we ended up with 1,600 more than 1,600 samples for barcoding. Here's another view of the whole room, and there's the voucher committee up there doing their job and all the mushrooms out there. So the way that we tracked our specimens is we use these tags. These were printed out by the VCP, include a QR code, which codes for this unique sample number, a place for the collector to put their name, a place for the foray location to be put. All these numbers can be connected to a location, and then a place to put an iNaturalist number if there was one. So the person doing the sampling, they would go around with the toothpick and the buffer, and they would dip the toothpick in the mushroom, dip it in the buffer, and then apply 2 stickers. One sticker pink, which highlights it as being sampled, and another white sticker that was easily removed. Then this would get photographed by someone who was taking photos either with their phone, or John Plischke had a full photography setup going. And the photographs were then uploaded to iNaturalist later. The folks that were sampling filled all this information out by hand on a piece of paper. Sigh. Also, sigh on having all the photos to bulk upload later. These were fundamental issues with the process, ones that have been improved since then in events currently. Everything that was sampled was also split to make a collection that went to the Mycota I think the Mycota fungarium is the largest private fungarium in the United States. I don't know. I'm pulling that number out of somewhere, but it's huge. There's like 100,000 collections there. A lot of them are tiny little splits just for the purpose of microscopy or DNA. Okay. After these things were sampled, we then set up PCR. So when we set up PCR, we do in batches of 96. So each of these sort of things with the foil wafer on, or the foil wrapper on it, have 96 little wells. And so we run PCR 96 at a time, and each well has its own unique index. And then each plate gets a secondary index to indicate which plate. So this plate has the index ONT 106. This has the index 104. And then each well has its own index. And these are short pieces of DNA that help us identify and connect a specimen to the DNA sequence. To make a run worthwhile, you have to do at least 480. And the run that we did on site was 672, which included all the specimens from the workshop class. Prior to the event, this is me setting up all the plates. So I had to add liquid to all these tubes. I have these fancy pipettes that do it, but this was the sort of prep process for that. And then after you do the PCR, you have to set up a sequencing library, and library referring to all the different barcodes combined together, that all of the individual indexes on them. It's a delicate process. When you make the library, you've got to also attach sequencing adapters, and every step requires a lot of focus. There's no room for error. If you make one mistake, the entire thing fails. Loading the flow cell, another delicate process. You may have seen that on the video. They add a little drop of liquid. There's a lot of things that can go wrong with that. So there's, it's a very nerve-wracking process, but once you get it going, you celebrate once you start seeing the good data coming out of there. And so we celebrated. Then you go wait 24 hours for the sequencing to complete. So the next thing you do is obsessively check your sequencing run throughout the night. Then you dream about your sequencing run and you sleep poorly. But this is good. That little peak that you see there means good. Then you wake up tired, you pack up your gear, and you throw the sequencer and the computer in the van till you head out to the next event. And then you have to stop somewhere and enter all that handwritten data into a spreadsheet. Never doing that again. That took a long time to enter more than 1,600 things into a spreadsheet. You run a software pipeline that then attaches the digital sequences to the index that you put together, and you upload it to a website called mycomap.com, which does the downstream analysis and prepares it for sequence validation. Voila. And that's it for NAMA 2025 at at Vermont. And these are folks from Myco, Quebec, who found some of the cool stuff that I'll show you in a little bit. Okay, I'm going to speed through these because we're already an hour in, so I've got a few more things to talk about. Continue doing PCR in the van. Okay, so here's the summary of results for NAMA 2025. We analyzed so far about 1,000 sequences with a success rate of only 78%. That's a little bit lower than I was hoping for. I'll figure out why it was so low once we get a chance to analyze all the data. Now, in Randall, Washington, we only had, 9 new state records? Vermont, we had 344 so far out of 1,000. That's a third of the things that we've looked at are new. Or sorry, even more than a third, because we analyzed 1,000, only 80% of them were successful. So, almost half the things that we analyzed are new state records. Now, when I say a new state record, I don't mean a new record of the mushroom for the state, a new record of the sequence. So, temper your enthusiasm. These are just new sequences, new sequenced records for the state. Never before sequenced taxa, however, was 46. So these are potentially new species. So compared to 19 or 18 from Washington, we got 46 with a new sequence discovery rate of 5.4%, which is pretty impressive. My validation crew, MVP for that was Tony Doucette from New Brunswick, Canada, 474. She really pulled that together. She carried the team. Runner-up Kevin Young from Indiana, 161. Garrett Taylor from New York, 124. Andrew, also from New Brunswick, 101. And Shannon Nix from New York, 23. So these are the folks that did the bulk of this analysis. So you should thank them. Now, what I'd like to do is show you some of the records that we found, and this would be a great opportunity for a couple more quick questions. If there are no quick questions, then I will continue.
There was a question Jessica may have answered already about at what point do you declare a new species? What's the differentiation?
Well, so once you have these, new sequences, there's quite a bit of work that needs to be done to actually declare something a new species. You'd have to hopefully link up with someone who's an expert in that group. Look through a lot of old publications, try to see if someone's already described it, but you just don't have a sequence for the holotype yet. Then you have to try to find the thing that you think it might be, get a collection of it from a museum, which is very challenging, sequence the holotype, successfully get a sequence from it, and then compare. In some cases where the holotype has been destroyed or is just irretrievable, you can use other sort of strong lines of evidence to rule out that it could possibly be that, whether it's host geographies, phenology, and of course microscopic characters. So to actually declare something a new species and describe it is considerably more work than it is. I get to do the easy part, which is just sequencing. The hard part is really describing new species.
And there was one other question that was sort of technical in nature about the 96 well plates and combining all those samples into a single sample, or exactly what is the situation?
Yep. So when I do PCR, I attach a unique index to all the samples and then combine them together before the library is put together. So when I do the sequencing, that index shows up and the computer reads it and says, oh, okay, this sample, this DNA sequence came from this well of a 96 well plate and came from this 96 well plate. So that's how we can mix everything together and sort it out later on.
Just got another quick question from Dawn asking about the toothpick method, and then is there a way to maybe resequence failed specimens from the toothpicks? I think is the question.
Yeah, so from the toothpicks, no, but from the buffer, yes. So I actually have all of the samples from every event I've ever done in my freezer at home. So that when I do the PCR, um, so I fill the tube with 30 microliters of buffer. For the PCR, I only take half a microliter. So in theory, I could do 60 repeats on one sample. And then Stephen Russell at Mycota has physical samples of everything else. So maybe the DNA extraction didn't go well. We can always go back to that. And that's in fact what we did in 2024. So the additional 400 sequences were stuff that failed the initial round or never got sequenced the first time. So I'm a little bit slow in getting through 2025 because I had a lot of events this year. But when we do get to the end of what I have already sequenced, whatever 1600 plus records, Stephen will pick up where, what falls through the cracks on my end, and we'll try to get as much saturation from the event as possible.
And two more quick questions that are just popping in here. Barati says, what is considered a good percentage of sequencing data? And the last one real quick was, how long does it take for the nanopore unit to do sequencing, from Scott?
Great. I'm assuming Barati's talking about success rate. So when I'm doing sequencing at home for my company, assuming homogeneous taxa, so not like biased on one taxon or another, I usually see between 90 and 95% success rate. I guarantee 75%. But 90 to 95%. It's a little bit different doing things with volunteers that may not have been trained on the sampling as well in, the chaos of a foray environment. So hopefully that answered that. And then it takes up to 24 hours to run the sequencing. If the sequencing is really good and the flow cell is high quality, I can usually get enough data to run my analysis after, 12 hours or even less. I think the shortest I've done was in Hawaii, where I let it run for 8 hours or 9 hours, and then it had enough data. So cool. So we're after 6. So I'm gonna jump into interactive mode, and we'll take a look at some of the collections we found, and then I'm happy to stick around for more stuff and more questions, but I want to make sure that I'm respectful of everyone's time. So, let's see. So just to quickly show you what our sequence analysis process looks like, and this is going to change. We're actually working with Mycota to develop a less clunky system. We put everything in these spreadsheets, and then whoever wants to work on them, of course, I'm showing myself at the top. Really, I didn't look at very many of these. So don't take that as me doing a lot of work on this. We can note the novelty. Was there a match greater than 99%? Have we uploaded to INAT? Is the analysis complete? That kind of thing. And then what I've done is I've filtered everything into a single sheet with just the stuff that is regional novelty, new to the universe. So new stuff. And without fishing through that, I've pulled aside some. Oh, this is our iNaturalist project, by the way. No need to get in the weeds on that. So here's some cool stuff that we found. So not just limited to really tiny stuff. Here's a great Arenia from Richard. So Tony did a great analysis on this one. And this mushroom is really cool. So we see this happen a lot where you get 2 sequences from one mushroom. And I'm not talking about like a lobster mushroom where you get an ascomycete and a basidiomycete. You get 2 Arenia sequences from one mushroom. That's kind of interesting. Sometimes they differ significantly. But what we can do is we can create a consensus sequence. So that's what this is here. So that's collapsing the variation between the two sequences into these what are called IUPAC ambiguity codes. And then we provide the individual sequence variants, or at least the common sequence variants. Sometimes you can have more than two in a single mushroom. You can have, 5, 6, 10. But these are the ones that occur, greater than 25% within that group of sequences. And this one was like the closest match was 10% away, or 17% away. That's really far. That's like really, really, really far. And so if you go to your iNaturalist observation, you can actually see how the analysis was done. You can click this link, and that brings you to the, we call them mycoblast results. And this is how we look at the how we compare sequences. So here was the top match here, 83%. Here's another one. So usually you call, if I were to pull a number out of thin air and say, what's the boundary between two species? I'd say at the 1% threshold. So if you have things that are 99% similar to each other, you probably have about a 50% chance that that's a different species. At the 2% threshold, I'd say you probably have 85, 90% chance it's a different species. At the 17% threshold, or whatever this is, it might even be its own genus. No way to know for sure with just one marker. Oh, it looks like I didn't look at the lower column of results. It does look like there is a match here that's within 2% to something found in Norway. Very cool. But still, but within that threshold of almost certainly a new species. Here's this Arenia, another Arenia. This was found by Jessica. You want to find new stuff, go for the stuff that's so small that you can't even get a picture of it. Is there a better? Yeah, here's a better shot of it. Yeah, this one. It's probably matches this thing from Montana, but everything else was 5% away. So we created this name Vermont 01. Even though it probably matches this thing from Montana, no one had applied a name to this thing yet. So this was just probably identified as, Arenia species. So we went and we just decided to give it a temporary name so that now anytime someone sequences something that matches this, we can give it that same temporary name. But 5% away from everything else, that's almost certainly not a new species, but a new sequence. Here's one by Kara Coulter. This one, the jury is still kind of out. Family Melissaceae. This would be a real odd one. This was identified as Calycina, which is what I would identify this as, morphologically speaking, because it's got this orange color. Melissaceae you almost always see as sort of a steely gray or bluish, greenish tone, occasionally some yellow tones, but this is really unusual. However, it does have this sort of wavy margin, and I'm not an ascomycete expert, so I could be totally talking out of somewhere, but I could see it potentially being in Melissaceae. Kevin took a look at this, and the closest matches were 98% Niptera raminicola. But these are, identifying these ascomycete cups is always a challenge. So you can't really trust the names that people use in their sequences. So that's another thing you got to be aware of, is that anyone can upload these sequences. I could upload a sequence of this and call it a giraffe. So you got to take everything with a grain of salt. And that's why we're being conservative and just saying, we're going to call it Melissaceae species, or Melissaceae species Vermont 01. But this could turn out to just be something really odd going on. Yeah, jury's out on that one. Here's another one from Caraculture, Henningsomyces. These are always fun to find. They're really cute. And I can't ask because you guys can't talk, but internal poll, ascomycete or basidiomycete? And just think about it for a second. The answer might surprise you. So it is in the order Agaricales. It is a basidiomycete. It's a Cyphalaceae, I believe. Don't quote me on that. So really tiny mushroom, really cool, and there's a ton of cryptic diversity. You know, identifying these things just by looking at them as a species, not easy. But with the DNA barcode, we can tell that this is pretty different from the closest match, you know, about 2% away. And that had a holotype, and that's Hengduanensis. That sounds like it's something from China. Let's take a look. And I can click this link and say, so HMAS, that's an herbarium code for Hong Kong, I think. And it doesn't say where it was collected. Oh, here we go. It doesn't say where it was collected, but it was submitted. So that's pretty cool. Something that's 2% away from something that was described from China. Here's Alora. Alora's running a really cool project in cahoots with Stephen Russell and Mycomap in British Columbia. Here's an inosobe. So this is more larger, sort of charismatic mushroom-ish. Inosobe, there's a ton of cryptic diversity. So this one, yeah, about 4% away from something called Inosobe assimilata. That's pretty far for Inosobe. Definitely distinct from Inosobe assimilata at that threshold. Of course, there's a little bit more than just looking at the percentages. I'm not going to get into that, but you can ask questions later. And then I'll finish up by showing this one. This is from Francois Guy from Montreal, from Myco Quebec, Mycosphaera. And Francois shows up to forays with knee pads on. So that's how you know that it's going to get serious. And so this thing, what a cool looking mushroom. Somewhere there's going to be a scale bar. So this is where we get, oh yeah, here, 100 micrometers. So like, and what is a hair, 50 micrometers in width? So this is like twice the width of a human hair, extremely tiny. And look at that cool blue coloring. And we're at the point where the macro photography with a camera and micro photography with a microscope overlap. That scale bar is the same size. You know, so we're getting into some really extreme macro photography. I think this was found in a piece of wet wood or something like that. And this one, I don't know how, but we did manage to get a sequence from it, even though how small this was. I think Francois used the tweezers under the microscope to sample it. And we have Keith Seifert weighing in on it. Keith is a prominent mycetologist. But there's a whole lot of discussion here. I'm not going to go into it, but it looks like we settled on Tubuiaceae. So really cool. This is not something that you'd be able to dry and then sequence later. It's just too tiny. So the only way we can discover stuff like this with sequence data is to be able to do this on site. So I don't want to run too far on time. I have a couple more things. God, there's so much to talk about. I thought I'd just show this one from this really cool Ramaria. And then I promised Garrett that I would go over a couple of his. Let's see, we got a Pluteus. Oh, maybe it's not showing up. Anyway, there was a really cool new Pluteus that Garrett found and a Milano as well. Those were just found in the past few days. So really great stuff. I'm gonna get back over to the slides. the rest of the slides I have are basically just showing off my adventures. That's not what I want to do. I'm gonna click this button. So I'll just quickly run through it. So after I did NAMA, did NEMF, upstate New York. Here's where we're doing PCR in in a room that had screens. There weren't even windows, and the screens were covered in dust, and there was like chipmunks running around. So for someone who asked about contamination, if there was a place that I could contaminate my samples, this would be it. And we saw pretty good results. Nothing showed up like Cladosporium or Aspergillus or any weird molds. So I think this process is pretty robust. Here's another NAMA event we did in Oregon Dunes. This one was really cool. Much smaller scale. We only did about 200 sequences. We found one new sequence and half a dozen state records. So pretty cool. Then I went to Northern California with the Fundus crew. We did a really cool, sampled some really rare plant habitat and did toothpick sampling here. You can see us at work. Then I did Olympic Peninsula Fungi Festival in Washington with Graham Steinruck and Biodiversity Collective. There's Chris Mettery, used to be a prominent player in the Telluride universe, learning how to do PCR. And that's it. That's all I got for you. So if you didn't get that link to the slides, there it is. If you can't click the links in the slides, I'm going to make sure that all those links are in the link tree. And so I'll open it up to folks for questions if you still have time for that.
Yeah, thanks, Hart. That was awesome. What a great presentation. And so far, like everybody's sticking around and watching it. So very entertaining and informative. There are some questions in here, and I imagine some more will come in. Let's take a look at them. Ari asks, would submitting fungi that are more than 1% divergent from a holotype be fast-tracked as a new species if it is shared with a taxonomist?
The term fast-tracked, are we talking about decades, hundreds of years, maybe? So I didn't get a chance to show everything off, but maybe I'll just quickly pop it open if I can do so quickly. I will try to do it. Okay. Well, this was Stephen Russell's dashboard, and let me see if I can find it. Species analysis. Okay. So hopefully this will populate in a second. All right. So fast track. Because of North American sequencing efforts, we have discovered 15,691 unique taxa as based on the molecular barcode. So again, we're not calling them all distinct species yet. We've created 8,700 of those temporary names. So that's a lot of stuff in the pipeline. Of those, over 7,000 only have a single collection. Very rare. Of these 8,700, 4,500 of them only have 2 collections. So fast tracking it would be if you're a taxonomist and you have a microscope and you want to write it up. And good news for you, there's a gentleman who might even be on this call who I've spoken about, Scott Ostoony, who's running a amateur publishing working group. And so if there's something you want to work up and describe as a new species, you don't need to forward it to a taxonomist. You can become the taxonomist. I don't have a link to these meetings or an intake form or anything like that, but perhaps we can work it out through the mycelium that you get a chance to attend one of these meetings. And if you are really dedicated to wanting to describe a new species, you'll be working with expert taxonomists during these meetings, folks like Alfredo Justo and Django Grootmeyers and others who actively describe species. And you can learn to do it on your own because this is a huge need. We need people to be excited about describing species. Doing the barcoding is the fun part. It's the hard work of actually describing new species that is required. So hopefully that answers your question.
Yep, and there's still a few more here. Manitoba Mushroom Society asks, how do we go about getting species submitted for sampling? I have some entered in INAT with dried samples available. No other sequencing being done in this area yet.
Great. Well, so let's see, micromap.org. Maybe this is the right place to go. So, Micromap Network. So if you go to this website, I think you can see current projects, find your region. So all of these regions here, you can submit samples to Mycota Lab for free sequencing. And then I think twice or more a year, even if you're not from these regions, you can submit samples to Mycota for free sequencing. And so, but that would be limited to, I think, 10 or something like that. I would refer to Mycota on the exact parameters. But if you really want your region to be involved, this is Manitoba. Oh God, I'm such an American. Forgive me. If you want your region to be involved and it's not already on this map, reach out to Stephen. Maybe you and your local myco club can raise some funds to set up a project. Alternatively, I do run a company that does DNA barcoding. And so if you have a bunch of specimens that you're just itching to get sequences for, my company is called Dicaria LLC, and the website is dicaria.llc. And that is as much of a pitch as I will give to my company.
One, one
let's see,
quickie
got that
here.
one answered. Thank you. Is it easy to distinguish whether a mushroom is edible or poisonous using DNA sequencing?
I'm going to say it's no. You can suspect that things are poisonous. So if you sequence something and it comes back closely matching something like Galerina, or if it comes back in Amanita section phalloides, you can suspect if it's a new species, right? And we don't have any data on it, you can suspect it's probably toxic, but you can't sequence something and know if it's non-toxic because there are so many fungal toxins. There's no way to know from a DNA barcode if a mushroom is edible or not, unless it matches something that you know is edible. And to be honest with you, learning your mushrooms, learn to identify them morphologically in person, I really recommend doing that before trying to sequence it, because by the time you get the sequence back, that mushroom in your fridge is not going to be good anymore.
Thank you. Sarah asks, has there been any attempts to combine soil DNA analysis with the fungal barcoding to try to get a broader perspective on the community at that location?
Yes. So I know Stephen Russell for his PhD project. You'll have to ask him the specifics, but I think he's done some soil sequencing in Indiana. And in Indiana, he's also done a ton of barcoding. And so those data sets overlap with each other. And so in theory, what you can do with that soil data is you can look and you can, put the sequences through an automated identification pipeline that does an okay job at assigning taxonomy to them. So let's say you've sequenced 16,000 collections from Indiana, and you're not generating any new sequences, or at least it's very rare that you get a new sequence. But you get some soil sequencing results back, and you see there's a DNA barcode from the soil that doesn't match your data set of physical collections that you've sequenced. Well, it might be a good idea to go back to that location and try to survey it again and see if you can find that specimen doesn't occur in your data set of sequenced specimens. In California, there's been a large soil sequencing effort. the biggest problem with soil sequencing is managing the data. The sequencing technologies that are used to generate it can sometimes lead to unreliable results. And just managing the amount of data you get from soil sequencing is a challenge. So, from one soil sample, you'll probably get thousands of species of fungi, maybe more. So, yes, the answer is yes, but how is another question.
Okay, I gotta let you know that in the chat, I think it was Chad said your logo is metal.
Thank you. Yes, my friend Sauce designed it, and she's great. So, yeah, I love it. And it's actually based on a culture that I grew when I worked with Alan Rockefeller, a culture of Psilocybe ovoideo cystidia.
Awesome. There's one more question in here. How about if I'll throw that out and then we can wrap it for the evening?
Great.
Okay, well, this has been great. Again, thank you. Um, Barati asks, or it says, new sequences are added to GenBank all the time, and literature is also updated to reflect current interpretations of phylogeny. What is the current practice to keep sequence validation current?
Um, I'm disentangling that a little bit in my mind here. So to keep it current, well, the landscape is constantly changing. The software that we use to do the analysis is improving. The platforms that we do the analysis on, like MycoMap, are improving. And the data is constantly flowing in. So, I'd like to say that we're getting more precise over time as more data gets added to these databases. MycoMap ingests the data from NCBI GenBank, constantly ingest the data from iNaturalist. So we will always have access to the freshest data, at least within iNaturalist and NCBI GenBank. Other databases like Unite and Bold take a little bit longer to seep in. But, as we go through, on MycoMap, there's some features. Maybe I can just show it really quickly. So within MycoMap, all these blast results are saved on the website, and we can edit them. So we can constantly be annotating the databases and changing, ignore Asterium, that's a mistake, but we can be adding our own annotations to these things as we come across them. And the more people are involved in this process, the more eyes you have looking at this, the more we'll keep these databases current. And hopefully that answers your question.
All right, well, thank you. I hope it does, because we'll probably need to wrap it up here. So if you want to say good night, it looks like people are in the chat saying good evening.
Yeah, thank you all so much for coming. And, I encourage you all to read through the documentation for the volunteer sequence group. Catch up with me on my socials. I think I got my Instagram link on the link tree. I'll pop that up for one more half a second in case anyone needs to quickly scan it with their phone.
Yep, scan that QR code right there, right?
Yeah, and if you want to get in touch with me, just message me on Instagram. My email is where messages go to die. So it's been great. Thank you so much, Trent.
Thank you, and thanks for all you do for NAMA. We really appreciate it, and we're looking forward to lots of future collaborations.
Great. Thank you so much.
All right, good night.